Experiment 10. Observation of permanent slide of plant cell. The aim of the experiment that we are going to perform today is to observe a plant cell under the microscope using a permanent slide. The materials required to perform this experiment are a permanent slide of onion peeling, a compound microscope, and a chart showing the structure of a plant cell as seen under an electron microscope. Method: place the permanent slide of onion peeling on the stage of a compound microscope. Focus the slide first under low magnification power and then under high magnification power of a compound microscope. Teacher's Practical Handbook. Come, let us study some tips from Teacher's Practical Handbook for this experiment. In case a permanent slide of onion peeling is not available, prepare a temporary slide of onion peeling following the procedure given below. 1. Take a small piece of onion peeling and put it in a watch glass containing water. 2. Take a clean glass slide and put a drop of water on it. 3. Transfer the onion peeling on the slide with the help of a brush. 4. Put a drop of dilute iodine solution on the peel. Cover it with a cover slip. 5. Wipe the excess of water from the sides of the cover slip with a blotting paper. This is a temporary mount of onion peeling. Observations. When we observe the permanent slide of onion peeling under the high magnification of a compound microscope, we see rectangular cells bearing cell wall are attached to each other. In these cells, dark-colored nucleus is clearly visible, which is present at the periphery of the cytoplasm. Cell membrane and lightly colored large plant vacuole is also seen. It is not possible to observe all the cell organelles of a plant cell using a compound microscope. Therefore, come, let us now study the chart showing the structure of a plant cell as seen under an electron microscope. In a plant cell, along with the plasma membrane, a rigid outer covering called a cell wall is present. Cell organelles like vacuole, mitochondrion, Golgi body, chloroplast, and nucleus are embedded in the cytoplasm of a plant cell. Come, after studying about the structural details of a plant cell, let us draw a well-labeled diagram of a plant cell. Observation of permanent slide of animal cell. The aim of the experiment that we are going to perform today is to observe an animal cell under a microscope using a permanent slide. The materials required to perform this experiment are a permanent slide of animal cell, a compound microscope, and a chart showing structure of an animal cell as seen under an electron microscope. Teacher's Practical Handbook. Come, let us study some tips from Teacher's Practical Handbook for this experiment. In case a permanent slide of an animal cell is not available, then prepare a temporary slide of an animal cell following the procedure given below. 1. Take a clean glass slide with a drop of water. 2. Using a cotton swab, gently scrub the inner surface of your cheek. The material, which is slimy, gets stuck to the swab. 3. Transfer this material on a glass slide. 4. Put a drop of methylene blue stain on the smear and cover the smear with a cover slip. This is a temporary mount of an animal cell. Method: place the permanent slide of animal cell on the stage of a compound microscope. Focus the slide first under low magnification power and then under high magnification power of a compound microscope. Observations. When we observe the permanent slide of animal cell under high magnification power of a compound microscope, we see small circular cells which are surrounded by a cell membrane. These cells have a dense cytoplasm. A darkly stained blue spot is also observed in the center of each cell. This is the nucleus of the cell. It is not possible to observe all the cell organelles of an animal cell using a compound microscope. Therefore, come, let us now study the chart showing the structure of an animal cell as seen under an electron microscope. In an animal cell, cell membrane forms the outermost covering of the cell. Cell organelles like vacuole, mitochondrion, Golgi body, and nucleus are embedded in the cytoplasm of the cell. Come, after studying about the structural details of an animal cell, let us draw a well-labeled diagram of an animal cell. All about cells. Every living organism is made up of cells. Cells are of various types. The cellular structure changes according to the nature of the work they perform. Plant cell and animal cell are different from each other in some aspects, but their basic structure is the same. Every type of a cell has a central controlling nucleus. The nucleus regulates all the functions of the cell. In the nucleus, there are chromosomes. Chromosomes are made up of deoxyribonucleic acid molecules. Chromosomes are bearers of hereditary characters and they transmit the genetic information from parents to the offspring. The genes present in chromosomes undertake the function of protein synthesis in the cell. Plant cell has nucleus, Golgi bodies, chloroplast, mitochondria, endoplasmic reticulum, ribosomes, and large-sized vacuoles. All these cell organelles are embedded in the cytoplasm of the cell. Cytoplasm is bounded by a cell membrane followed by a cell wall. Chloroplasts, which contain chlorophyll pigments, carry out the function of photosynthesis, and hence only plant cells are capable of the photosynthetic activity. In animal cell, cell wall and chloroplast are absent. Cell organelles called lysosomes are present.